Hello, everyone. I'm Jim Eubanks. I'm coming to you from a soon-to-be stormy Toronto, Ontario, Canada. And I'm just going to change over and start my slides. Just give me 1 second. And I'd like to thank the organizers for giving me the opportunity to present some of the work that we've been doing towards the generation of a personalized HNRNPH2 deficiency disorder mouse model, hopefully to allow us to elucidate pathogenic mechanisms better and streamline the development for effective therapies. As I mentioned, my name is James Eubanks. I'm the division head for experimental and translational neuroscience at the Krembil Research Institute in the Toronto area. And I'm also a professor in the Department of Surgery at the University of Toronto. Let me also mention up front that I don't have any conflicts to declare. So as everyone is all likely very well aware, HNRNPH2 mutations cause a neurodevelopmental condition. And as shown here, this condition can affect a number of different areas of the brain and body. It's associated with a spectrum of different affectedness, developmental delay, craniofacial alterations, limited hand use, limited speech. In some patients, seizures have been identified, poor general sleep patterns and hypotonia and perhaps bone abnormalities have all been identified in different patients. But patients do have a range of affectedness, and many or only a few of these different individual phenotypes may be present in any one given individual. Now, as I mentioned, mutations of this gene, which is X-linked, are causal for the condition. And in this cartoon, we see a schematic of the X chromosome and where on the X chromosome the gene for HNRNPH2 resides. It resides on the long arm of the X chromosome, shown here. Structurally, it has a fairly simple structure. It has two primary exons, shown here in green in an illustration of the gene structure. There is the one intron that separates the two exons. And interestingly, all of the open reading frame that encodes for the translated protein that becomes the functional HNRNPH2 protein is included in this one exon 2. Now, although this is on the X chromosome, the HNRNPH2 gene is really not near either the gene of MECP2, which is at the very terminal tail end of the X chromosome on the long arm, and when mutated causes the other neurodevelopmental condition of Rett syndrome, or CDKL5, which is located far removed and on the short arm of the X chromosome, where mutations cause the CDKL5 deficiency disorder. So this just illustrates that HNRNPH2 mutations are very distinct, and causally, they're very, very far removed from the mutations that would cause either Rett syndrome or CDKL5 deficiency disorder. When one looks at the structure of the protein that's encoded, modelers have presented this as a likely structure for its three-dimensional shape. Within the structure, there are different known motifs that have high homology to areas that bind different types of RNA within the cell. And I just want to draw your attention to this area, which is the NLS, or the nuclear localization signal, which is a series or a sequence of amino acids that encode for a functional domain that brings the protein into the nucleus of the cell. This figure is modified from Jepson et al. Now, we know that there are different mutations of HNRNPH2 that do cause the condition, but two mutation sites within the encoded protein seem to be very common. And those are illustrated here with these two hatch marks. These are missense mutations where a single amino acid is changed. And in each case, it resides within this nuclear localization signal sequence. The most common pathogenic mutations seem to occur within this nuclear localization signal. The areas that are affected encode for either the normal amino acid at position 206, which normally is an arginine, or affects the normal amino acid position at 209, which is normally a protein. Now, at position 206, there's actually been different mutations identified that either change the arginine amino acid into a glutamine, so that would be the R206Q mutation, or change the arginine into a tryptophan, which would be the R206W. And these alterations likely affect the function of this nuclear localization signal and prevent or impair the ability of the protein to actually process mRNA properly within the cell. Now, my laboratory has been funded to study neurodevelopmental conditions for some time, and as part of our work with the Ontario Brain Institute, we've been funded to generate new mouse models and embryonic stem cell lines that genocopy rare genetic mutations that are seen in Ontario area patients. And there's one Southern Ontario patient that expresses an R206Q mutation of HNRNPH2. And so one of the mouse models that we've now generated is indeed this R206Q missense mutation of HNRNPH2. We used CRISPR-Cas9 targeted gene editing to introduce this mutation into mice. We did this in mouse embryos, and we received roughly 20 founders. So these would be the firstborn from our manipulations. And only one was identified that expressed the proper targeted mutation but lacked any off-target effects. And we did a whole battery of quality controls to confirm that we had modified the intended nucleotide sequence, but didn't introduce any other alterations into the genome. This founder was successfully bred, and this new HNRNPH2 R206Q line was established, and this is one picture of one of the mice. This is actually the original founder mouse that was born. However, in a way, we got a little bit lucky because our editing process also generated a different mutation at position 206, where the nucleotides had been changed to encode for a histidine amino acid rather than a glutamine. And we believe that this is due to the homologous recombination with our editing template occurring right between the G and the C, where the A would be from the engineered oligo for the recombination, but the G in the Q is from the targeted oligo, but the C in the H form is actually what was seen in the wild type gene. So the net result is that we actually got two different mouse lines, one expressing the Q that's seen in patients, and a histidine, which, like the arginine that's normally encoded, is positively charged at physiological pH, but certainly has a different structure than the arginine. But we also introduced the Q and the H mouse. Both of these mice are viable, they reproduce, and they show very few overt abnormalities. And this is just a picture illustrating the behavior of the Q mouse when placed on a podium, and also the 206H mouse. And there's nothing overtly wrong with either of the mice, at least at this high altitude view. They behave like mice, and they do most of the normal things that mice of this age would do. But we've also started to assess some behavioral phenotyping, and some symptoms in these mice are beginning to be observed as they mature. Now, I have to put the caveat note in here that these results are preliminary. They certainly have been hindered by our COVID shutdown, and we've only been able to conduct assays on mice that are 6 months of age or younger. And at this time, most of the work that we've done has focused on male mice. We do have the heterozygous female mice, which would be the better genocopy of what is present in most patients, but I don't have any data on the heterozygous females to share today. But I do have some data on the male mice. And the first thing that we noticed was that the R206Q mice, but not the R206H mice, show an abnormality in growth. They're at weaning smaller in mass, and they maintain this smaller than normal mass size all the way out to about 3 months of age. This was not seen in the H males. The 206H are normal at the weaning age, and their growth rate parallels that seen of the wild type mice. Interestingly, in roughly a little over half of the male R206Q mice that we've examined to date, but none of the R206H mice, but in the R206Q mice, we do see an abnormality in the craniofacial structure. They have a domed head, and that's very clearly seen here relative to a wild type littermate mouse that has the regular profile of the sloped head. And you can see the the dome shape in the cranium of the R206Q male mouse. Necropsy examination of this mouse and others has revealed that it's due to a hydrocephalus that's present. And interestingly, hydrocephalus has been observed in some of the HNRNPH2 mutation patients. And so this may phenocopy what's seen in at least some patients, albeit probably to a larger degree in the mouse than what's been seen to date in patients. We also notice that there's a hind limb clasp reflex impairment. It is seen in both the R206Q mice as well as the R206H mice. When you elevate a wild type mouse or a normal mouse by its tail, it anticipates that you're going to drop it, so it spreads its hind legs and its forelimbs to catch itself when you do drop it. That's the normal reflex, but that's impaired in both the R206Q and the R206H mice. They clasp their forelimbs, and it's more pronounced in the Q, as I mentioned, than in the H, but both do it to some extent, and their hind limbs don't get fully extended. And so this hind limb clasp impairment has been seen in other neurodevelopmental mouse models, certainly in the MECP2 Rett syndrome mice, as well as in the CDKL5 CDD mouse. And it may have some similarities to hand stereotypies that can be seen in some patients of any of those conditions. We also notice that the 206Q mice, but not the 206H mice, have a higher than normal risk for sudden and unexpected death. And this is the Kaplan-Meier survival probability for the male R206Q mice as a function of age, as well as the R206H mice, and it's over the same time windows. And the blue line shows the death rate, either found dead in their cage or reached a point where we had to euthanize the mice. And roughly 83% of the mice with the R206Q died before 20 weeks of age. We only saw one mouse herd, meaning that about 90% of the R206H mice survived longer than 30 weeks, and many of these mice are still going. So it illustrates that, at least in the males, the presence of the R206Q mutation does enhance the risk for sudden and unexpected death. So those were some of the overt characterizations that we saw, but we've also started to investigate some behavioral performance tasks. And like the general behaviors, these task results also suggest the presence of phenotypic impairments, and particularly in the R206Q mice. Again, I must highlight that the caveat being that the assays are still at their preliminary stage. They've only been conducted on a few mice, and all of whom who have been examined are less than 6 months of age. But when we look at some of the behavioral performances, we do start to see some tendencies. We first examined anxiety-like behavior using this elevated plus maze apparatus. This is a platform that is elevated above the floor by about 3 feet. It has one arm that's open and then one arm that's closed. Normally, mice don't like to stay out in the open. They're a little bit scared that the hawk may come and find them. And so they tend to spend most of their time in the closed or the safe arm of the maze. But they're also curious and exploratory creatures, so they do occasionally come out and explore. And so shown here would be the normal amount of time that a male wild-type mouse would spend in the open arm over a 5-minute period. And you can see very clearly that the R206Q null mouse spent considerably longer in the open arm relative to the wild type. However, the 206H mouse performed identical to wild type. And so this suggests that the R206Q has a diminished anxiety-like phenotypic behavior, but not the R206H mouse. For females, this is not yet far enough along to be confident of what the result is, but at least at this stage, we didn't see the same tendency. There was no significant difference in the female H or the female Q mice relative to what a wild-type mouse performance was. But more investigations in the females, as I mentioned, will be coming. We also use the light-dark place preference test to assay anxiety-like behavior. It's a similar assay in that it has a light compartment and then a dark compartment that's allowed by a door for the mouse to go back and forth freely. We illuminate this with a bright light, and like before, most normal mice would prefer to stay in the dark area where it's safe, and that's shown by the time that the wild-type mouse spends in the light. And like in the elevated plus phase, we saw a significant increase in the amount of time that the R206Q mouse spent within the light chamber, but no difference was seen in the R206H mouse. Again, female mice didn't show any effect, at least at the stage of development that we investigated. So these results together would suggest that the presence of the R206Q form of HNR-NPH2 either decreases the normal anxiety-like behavior, they become more exploratory or more curious of their environment relative to what the wild-type mouse is, or the histidine R206H mutation. When we looked at their open field exploration, however, we got a little bit of a surprise, and this measures both their activity, their exploratory interest, and their locomotive ability. And what we do is we put the mouse in just a simple open field box and let it explore around. And what we found is over a 1 hour period, there was no significant difference in the total activity of the HNR-NPH2Q mice or H mice relative to wild type. No effect was seen either in any of the female mice that were examined. So this was a little bit of a surprise in that we thought that there might be a decrease in the total activity, but the assay didn't support that prediction. Likewise, locomotive activity, this counts how far the mouse will walk over the 1 hour period within the open field apparatus. Again, we didn't see any significant effect in either the R206H or the R206Q mice relative to wild type. They walked the same amount of distance, and the same was seen in the female mice that we also examined. So these results, while preliminary, I acknowledge up front, they do suggest that locomotive activity is preserved in both male and female R206Q as well as R206H mice, at least until 6 months of age, and we'll be following this in a longitudinal manner as the mice age. So I'd like to summarize here by saying that we have now generated 2 different lines of HNR-NPH2 mutant mice. One expresses the R206Q mutation that is seen in patients, and one expresses an R206H mutation that to date has not yet been observed in patients. The data that we have to date suggests that this R206Q mutation in mice has a greater impact on both general performances as well as the mouse performance in some of the behavioral tests that we employed, while the 206H mutation doesn't seem to have nearly the same impact. We've seen some impairments in the R206Q mice that may recapitulate some of the impairments seen in patients, such as the hydrocephaly, the association with the hand stereotypies, the curiosity or increased curiosity behavior, aspects like that. They may be showing up in our R206Q mutants. But we also found some behavioral features in these R206Q mice that don't seem to mirror the features that are commonly seen in patients. And for example, the preserved mobility and ambulation would be examples of this. I will say clearly that more work needs to be done in all of these areas, and we're in the process of conducting additional assays. Our lab is now reopened from COVID, and this is one of the priorities that's going on. So we think that this is an advancement in the field, and that the availability of these personalized HNR-NPH2 mouse lines that do appear to show some phenotypic similarities to the patients will aid in delineating the pathogenic mechanisms that are associated with this condition, and in doing so will provide a valuable resource for finding and developing new treatments that will effectively improve the quality of life for the patients with this condition. And I'll just close by acknowledging the people in my laboratory who conducted the work. This is a picture of the Krembil Research Institute at our site. And I'd like to acknowledge the people, the Krembil Neuroscience Center, as well as the Ontario Brain Institute that provided the funding for our work. And with that, I thank you for your attention, and I'll be happy to answer questions.
Amazing.